Activation of GQ in endothelial cells lining blood vessels leads to nitric oxide release NO and blood vessel dilation. Acetylcholine receptors on endothelial cells lining blood vessels are G-protein coupled receptors. Acetylcholine binding to its receptor leading to GQ activation. The activated G-beta gamma subunit of GQ binds and activates PLC-beta. PLC-beta activation leads to IP3 production from PIP2. Increases in cytosolic IP3 levels lead to a cytosolic calcium spike. Calcium activates NO synthase, or NOS, and activated NOS produces NO from arginine. NO is a small nonpolar molecule which can easily diffuse out of endothelial cells. NO diffuses into smooth muscle cells, also lining blood vessels. NO binds to and activates guanylyl cyclase in smooth muscle cells. An activated guanylyl cyclase catalyzes the transformation of GTP to cyclic GMP. Cyclic GMP is a second messenger. An increase in cytosolic cyclic GMP concentration leads to smooth muscle cell relaxation and a decrease in blood pressure. The decrease in blood pressure can be local or systemic if pharmaceuticals are used. And we have a picture here. The main thing about the signal transduction pathway, it is activated in one cell, and then its response is actually in another cell. You have the endothelial cell, and the endothelial cells are right here. So here's the blood vessel, right? So here's our endothelial cells. You can see there's 4 of them in this picture, and let me get rid of this right now. And you can see here's the basement membrane. Just the way we said it before. Endothelial cells are going to be bound to a basement membrane. And you have the lumen of the blood vessel right here where the blood is going to be, right? And then you have the smooth muscle cells right here. And you can see them right here. Here's one of them, right? On the other side of the basement membrane. And the basal lamina just being the basement membrane. Now the signal will start right here in the endothelial cells, where acetylcholine will bind to G-protein coupled receptors on the surface of endothelial cells, and that will lead to activation of GQ, which will lead to IP3 production, which will lead to everything that we talked about previously with the PKC pathway. But instead, when calcium levels go up, another thing happens where NO is made by NO synthase using arginine. NO is a small nonpolar molecule, and it will easily diffuse down its concentration gradient. So you will have a buildup of NO in the endothelial cells, and NO will diffuse out and will diffuse into an area of low concentration, for example, the smooth muscle cells, which have very little NO in them. NO will bind to guanylyl cyclase right here and activate it. That will lead to GTP being transformed to cyclic GMP. And cyclic GMP is a second messenger. And its action will be that the smooth muscle cells will relax. And if you have relaxation of the smooth muscle cells, that will lead to the lumen of the blood vessel increasing, so blood pressure will drop. Note that this is a paracrine cell-to-cell signaling, and that the actions of one cell type is under the control of another cell type. This is an example of a response being generated by the calcium spikes of the PKC pathway that is independent of PKC activity. The NO pathway can be written as follows. So we'll start out with the endothelial cells. And everything that's happening in the endothelial cells, we'll write it in blue. So we have acetylcholine activating the G-protein coupled receptor, which then goes on to activate GQ, which then goes on to activate PLC-beta, which then goes on to activate IP3. We're not worried about DAG in this pathway. It will be produced, but it's not important for us. This will go on to activate IP3 receptors. Now we're going to have the release of calcium from the ER. Now the positive feedback loop is there with ryanodine receptors, which leads to more calcium being released, but also you have the negative feedback loop of calcium on both these things. Now, what we're going to do is we're not going to put this in the pathway. I wrote it out so that you guys know that this hasn't disappeared, but we're not going to put it in because it'll make it a little bit too busy. So we're basically summarizing a little bit, but just be aware that that does still exist. Okay? Now, calcium release will lead to the activation of NO synthase. NO synthase will go ahead and produce NO from arginine. Now, at this point, you'll have diffusion, and we'll have diffusion in red. And where's it going to diffuse into? Into smooth muscle cells. Everything that's happening in the smooth muscle cell will write in green. So our NO that was produced in the endothelial cells will go on to activate guanylyl cyclase, and this will activate or produce cyclic GMP from GTP. And that will lead to the response. And what is the response? Smooth muscle relaxation. And smooth muscle relaxation will lead to lowering the blood pressure. Molecular mechanism for smelling, olfactory pathway. The molecule binds to an olfactory receptor, a G-protein coupled receptor, on a cilia of olfactory neurons. So here you can see that we have the cilia kind of looking like these hairs sticking out from these cells. They kind of behave like dendrites of a neuron. And you have G-protein coupled receptors on these cilia that will bind specifically to certain molecules. And that will initiate eventually an action potential that we fire down the axon. So the structure of an olfactory cell or olfactory neuron is very similar to the neurons we talked about before. You have the cell body right here. Or the soma. You have the axons right here that are being shown right here. And you have these modified cilia that kind of behave like the dendrites. And you have a picture of it right here, a real picture. Activation of the olfactory receptor, meaning the binding of whatever molecule that you're going to be smelling, leads to it binding its trimeric G protein, which is called G-olf. The activated G protein coupled receptor acts as a GEF and activates a G-alpha subunit of G-olf. An activated G-alpha binds and activates adenylyl cyclase. An activated adenylyl cyclase catalyzes a transformation of ATP to cyclic AMP, which leads to a cytosolic increase in cyclic AMP concentration. Cyclic AMP binds to cyclic AMP activated cation channels on the cell membrane, which leads to an influx of mostly sodium. So instead of having transmedicated channels, what you're going to have is an action happening through the G protein coupled receptor leading to the activation of adenylyl cyclase, which leads to an increase in cyclic AMP concentrations. And then cyclic AMP will bind to ligand-gated channels, where the ligand binding pockets are cytosolic. Filling them with cyclic AMP will lead to those channels opening, and those are cation channels. And cation channels, when you open them, they will mostly conduct sodium. Note, this is an example of a ligand-gated channels where the ligand pockets are intracellular. The influx of sodium into the cilia will lead to the depolarization of the cilia membrane, something we've seen before. If the combined PSP is strong enough, past threshold, an action potential will be generated and sent down the axon of the olfactory neuron. The signal is then passed on to neurons in the brain. The olfactory pathway can be shown as follows. Signaling molecule. In this case, the signaling molecule is whatever molecule that you're going to be smelling. Activating G-protein coupled receptor, which then goes on to activate G-olf, which then goes on to activate adenylyl cyclase, which then leads to an increase in cyclic AMP concentrations in the cytosol, which then leads to the activation of cyclic AMP activated cation channels. This will lead to the depolarization of the membrane, which will lead to an action potential being generated. And that is if the PSP is past threshold. And that's the olfactory pathway. That is how you smell. Okay, we'll stop here and continue later. Thank you, guys.